Hello, and welcome to the latest Technology Networks webinar, In Vitro Antibody Discovery and the Potential Value of AI, the AIntibody Challenge. I'm your moderator, Dr. Stephen Gibney, Science Writer for Technology Networks, and I'm excited to be here to host today's session. We have two fantastic presenters, Dr. Andrew Bradbury and Dr. Daniel Bedinger, who will share some of their valuable insights with us. Andrew is the Chief Scientific Officer of Specifica. He trained in medicine at the Universities of Oxford and London and received his PhD from the University of Cambridge at the MRC Laboratory of Molecular Biology. Driven by Specifica's mission, Andrew is committed to developing therapeutic antibodies with the world's best antibody selection platform. Daniel is Senior Manager at Cartera, who was instrumental in the launch of Cartera's LSA platform, perfecting technology, applications, and methods. He now leads the company's global application science team and has over 2 decades of experience in the generation and characterization of therapeutic monoclonal antibodies. Daniel earned his PhD from the University of California, Davis in cellular and molecular physiology. After their presentation, we'll have a short Q&A session, and we encourage you to submit your questions at any point during the presentation. To do so, simply type your question in the box on the right-hand side of the screen and click Send. We'll do our best to address as many questions in the time that we have available today. If you encounter any technical difficulties during the webinar, click the chat box on the right-hand side of your screen to request assistance from our support team. Without further introduction, I'll hand over to our speakers. Andrew, Daniel, over to you.
Thanks for the introduction, Stephen. So today what I'd like to talk about is sort of AI and antibody discovery and how it compares to experimental methods. The key objectives here are to understand what's really possible, provided a relatively straightforward challenge with lots of data available. And so the data that we're going to be talking about today is all based on SARS-CoV-2, and in particular the RBD, which is the most studied protein in history. The rationale was that if AI could not work with RBD, then it was probably unlikely to work with other things as well. We wanted to define where in discovery, antibody discovery, AI could be helpful, and determined if there were particular methods or groups that had a clear advantage. So the source of the data is based on Specifica's Gen 3 antibody discovery platform. Illustrated here is a guide to how the library is made and what the various components of the library are comprised of. And so you see here a light chain schematically indicated. All the light chain frameworks come from therapeutic scaffolds, and all the light chain CDRs come from a next-gen sequencing database that we have at Specifica, which has been purged of sequence liabilities and is germline matched to the therapeutic scaffold. In the case of the VH, it's similar but not the same in that the first 3 frameworks all come from therapeutic scaffolds, and VH and VL are matched. They both come from the same scaffold. CDRs 1 and 2 in the heavy chain also come from the same next-gen sequencing database, purged of sequence liabilities and matched the germline. CDR3, on the other hand, because of the great diversity of CDR3, cannot be synthesized, and these are derived by amplification from a unique set of 10 donors. So the platform we use to do selection, we build libraries that are about diversity of at least 10 billion different clones. We use a trillion phage per selection. We do a couple of rounds of phage selection, and the purpose of this is just to bring the diversity down enough to be functional within the context of yeast display. And within yeast display, that's when we are able to do fine-tuning of affinity, specificity, look for antibodies that bind or antagonize particular epitopes and the like. And then under normal circumstances, when we're doing a discovery campaign, we have enough diversity there to just need to pick 96 or maybe up to maximum 400 different clones, and we can get the diversity required then. However, in some cases, we also carry out next-gen sequencing and analysis, and that allows us to further expand the diversity, identify new clusters, and also identify new binders within particular clusters that have been described by clustering the HCDR3. Now, up until recently, it was impossible to compare discovery platforms. And the reason for this is because everybody was comparing the output of their discovery platforms against different targets, often using different antibody formats, and often testing different biological endpoints. Now, that all changed with COVID. This was in an inadvertent natural global experiment. Everybody who knew anything about antibodies was selecting antibodies against the same target, which was the receptor binding domain of SARS-CoV-2. Everybody was testing IgG, and everybody was looking at affinity, IC50, so that is the live or pseudoviral neutralization, and the recognition of variants. So if you look in the literature, there are hundreds of peer-reviewed publications describing antibodies against the RBD of SARS-CoV-2 now. So in 2021, Erica Ullman-Saphir's group published a paper where she described the properties of 180 antibodies from 56 different labs, derived using many different methods, including immune, naive libraries, in silico design, and the like, and all measured together in the same platform, which was SPR, surface plasmon resonance, and the Cartera LSA. So what we did was we used our platform to generate antibodies against the same target, and we measured 129 antibodies, which were derived from this selection and identified by next-gen sequencing. And these were tested also by SPR on the Cartera platform. Obviously, this was a machine in our facilities and not in the La Jolla Institute of Immunology, where Erica Ullman-Saphir works. And then in addition, we also took some additional antibodies, some of the really high affinity ones, and we tested their affinities by Kinexa. And so on the left, you see that basically 4 or 5 antibodies from these 180 had affinities which were better than 100 picomolar. Compare that to what we were able to derive. We found 40 antibodies with affinities of better than 100 picomolar, with the best affinity antibody straight out of the library without carrying out any affinity optimization was 13 picomolar. So this is the source data for the AI antibody challenge that I'm going to talk about in a minute. Now, this platform works extremely well, not just with SARS-CoV-2. Should mention this library was built before the pandemic, so it is not a SARS-CoV-2 immune library, but it's also been tested against a large number of other proteins and targets as indicated here. And just to summarize quickly, if you exclude peptide MHC, about 70% of campaigns generate sub-nanomolar binders, with about 35% having affinities better than 100 picomolar. About 20% of binders altogether are sub-nanomolar, and worst affinities come from selections that are more complex. So things like peptide MHC, which has a very small epitopic space for discovery, and then domain, isoform specific, or species cross-reactivity type selections. We've provided this library to many clients, as indicated here. Altogether, we've now delivered 26 platforms, and I think that's to about 14 or 15 different clients. Now, the question we're trying to address here today is, does AI make in vitro platforms redundant? And so I thought it would be interesting to ask ChatGPT what it thinks about AI in antibody discovery. So now, if you take this assessment and others as well, generated by other large language models, you'd come to the conclusion that AI is going to put us all out of business. Now, I was a bit skeptical of this, and so along with Frank Erasmus, we established the AI Antibody Challenge, and this was loosely inspired by CASP, which was a protein structural prediction competition, which was the first to identify AlphaFold as being such a powerful protein structural predicting algorithm. We organized this first competition very much ad hoc. Time was of the essence. We wanted to move quickly. We felt that the field was moving very quickly, and we didn't really have time to put together a committee and search for funding and the like. Everything was carried out blinded, and the competition was initiated in Q1 of this year. We made all the NGS datasets that I'm going to talk about available for downloading by participants simultaneously. They had 14 days to upload their designed or identified antibody sequences. And then we will be publishing a results manuscript, which is in the process of being, a first draft has been written, is being reviewed by some of the participants. And we're going to be publishing the results blinded except for the winners. And each participant will know their rank within the spread, but not identified. So the first challenge was an in silico antibody affinity maturation challenge. I'll say more about this, but we provide 3 data sets and ask the participants to design an antibody based on these 3 data sets with improved affinity. The next was an in silico affinity rank prediction. So provided with 3 HCDR3 clusters, identify the highest affinity antibodies in those clusters. So these are based on, this is NGS of an experimental output, and all the AI is being asked to do is just to predict which of the antibodies in that output have the best affinities. And finally, this was a challenge 3 was an NGS inspired computational antibody design. So given a data set of over 30,000 sequences with some affinities, design an out of library antibody sequence, changing only the CDRs, not touching the frameworks, and that binds the same target with the highest possible affinity and favorable developability properties. The announcement manuscript was published a year ago, indicated here. And altogether, we had 29 participants with over 500 submissions, broken down, as you can see here. The greatest number were non-profit or academic groups. AI biotech came next. And then there was a couple of categories of companies, either small, medium-sized biotech or large biotech pharma, that did not have AI at the center of their capabilities, but nevertheless wanted to participate. And you can see the number of antibodies provided for each of the 3 challenges on the right-hand side. And the full data set measured by Cartera was a total of over 500 antibodies, each measured in triplicate for a total of just under 1,600 different analyses. And you will have heard already or will be hearing about Cartera analysis. So how do we determine the ranking for these different challenges? So what we have bottom left here is an affinity range for antibodies and the best ways of measuring them. And you can see that the affinity is going from about 1 micromolar down to about 10 to 100 picomolar, best measured by SPR, in particular the LSA and Cartera. But if you have affinities better than 10 to 100 picomolar, then Kinexa is by far the best way of measuring these affinities. And I'll show some correlation data later. And so we received the submitted antibody sequences, provided them to Azenta that produced the antibodies, and then shipped those antibodies to Cartera for SPR measurements and Mosaic for developability. These were rank ordered by SPR. Poorly developable antibodies were eliminated, except for some of those on the borderline, and they were all considered for Kinexa. We first carried out a single point screening to confirm the consistent rank order compared to SPR, and then finally, the best antibodies, up to 5 candidates for each challenge, were a full Kinexa screening was carried out. So how did we work out the developability? We had 5 different developability assays indicated here: HIC, BVP, polyreactivity, AC signs, self-interaction, TM, and TAG. The passing levels are indicated here in this passing column, and the failing is indicated on the right-hand side, and questionable was between these 2 ranges. So if an antibody passed, it was given a score of 0. If it failed, it was given a score of 2. And if it was in between, it was given a score of 1. And in order to pass, the cumulative score across 5 different assays had to be 3 or less. And if it was 4 or more, the antibody failed. Now, if we're looking at the affinity, This illustrates now a comparison of the affinities measured by Kinexa on the y-axis, SPR by the LSA on the x-axis. And you can see while there's a very good rank order correlation between these two ways of measuring, the one-to-one affinity correspondence reference line shows that there is a pretty poor absolute correlation, particularly at high affinities. Kinexa generally gives much higher affinity values, which we believe, the field believes, are more realistic. And so Kinexa values were prioritized for high affinity antibodies. A similar correlation between single point Kinexa and SPR showed pretty much the same. The absolute correlation, again, was pretty poor. The rank order correlation, however, was quite good. And then comparing the Kinexa single point to the standard Kinexa, you can see again, now we're getting an excellent correlation, both rank order and absolute. So let's go to the challenges. So challenge 1, in silico antibody affinity maturation. So the way we carry out affinity maturation of specifica is we create libraries. We take the antibody, the parental antibody, we put libraries of different CDRs, all of which are natural, matched to the germline of the antibody, the affinity of which is to be improved, and purged of sequence liabilities. We make a library in L1, L2, another library in L3, another library in HCDR1 and HCDR2. We usually leave HCDR3 alone in order not to affect the binding properties, the epitope recognition of the antibody. And then we select outputs for each of these, and then these outputs are combined in a combination library illustrated here. And then on the basis of that, we carry out a further selection, and those are the affinity matured antibodies. And generally, experimentally, we are able to, we guarantee, in fact, an improvement of at least 10-fold. And the best improvement we've got has been almost 30,000-fold. Now, the challenge here was, given these 3 data sets, so that's a selection output where antibodies are improved for binding, where the diversity is L1, L2, or L3, or H1, H2, then design a full-length antibody using CDRs that is going to bind the target better than the parental. And so the experimental route, as I've just described, takes 2 to 3 weeks to make these 3 individual libraries. Once we've done the individual libraries, then the experimental route takes us an extra 2 to 3 weeks to build a combo library, to carry out a selection, and then to identify a panel of improved variants, another 2 to 3 weeks to produce IgGs, and then another 1 to 2 weeks to measure the affinity either by Cartera or by Kinexa, and also assess the developability. So altogether, this experimental approach is a 9 to 12-week process. Now, if you could eliminate that second portion, having generated this data here, if you could do artificial intelligence, so just a couple, say 3 days to identify improved antibodies, we'd still need to test them. So we still need 2 to 3 weeks for Azenta and IgG expression. We'd still need to measure their affinities and their developability. But altogether, this is now a 6.5 to 9 week process. So we're saving 2 to 3 weeks over the traditional experimental approach. Now, if one could just do in silico binding prediction immediately, that could be done in just 1 or 2 days. But combined with the so this is in other words, this is taking, not bothering with the first experimental phase, just taking the parental and doing in silico binding prediction. This would reduce the time to 5.5 to 7 weeks. And then finally, of course, if one could do just direct 0 shot, no target, no affinity required, which of course is where we'd all like to go, that could all be done in about a week. Now, what does the data look like? So this is the overall SPR measurements. So what we have here is all the parental here in red here. The best experimental is illustrated here. And what you can see is there's a very broad distribution with about 50% of submissions with some level of improvement. So all these round circles represents AI submitted antibodies, and you can see that All of these are improved over the parental. These are worse. These ones down here, these crosses, don't bind at all. The experimental approaches, all illustrated here, these blue X's, generally they almost all had improvements that were tenfold, at least tenfold over the parental. And there were some that were extremely high affinity, as illustrated here, down to about 9 picomolar. But as I mentioned, this takes 2 to 3 extra weeks. 31% of the AI submissions were non-binders. And there were 2 AI antibodies illustrated here, which had improved affinities over the best experimental and were also developable. So if we're looking at all the data in another way, then what we have here, this is the SPR data, this is the Connexa data. Unfortunately, the Connexa data is still being assessed, but what you have here is the best experimental threshold, illustrated here on the SPR. What you can see is that a couple of, and this is the parental, sorry, in blue, and you can see that 20 to 30% of the antibodies in the challenge have a 5 to 10-fold improvement over the parental. The top 2 organizations had very high affinities, as illustrated here. Only one of them beat the experimental by SPR, and this one here is slightly worse than the best experimental. But they all were developable. There's a third place antibody, which was unfortunately disqualified, and that's because it was not developable, as illustrated here in the purple. Now, we're also looking at the Connexa measurements. So the remaining experimental controls are being measured, as I said. The Connexa gave much better affinities to the improved, the AI, the best AI antibodies, with about a 2,000-fold improvement over the parental, with the first place antibody coming significantly better, 3-fold better than the best experimental. Okay, challenge 2. So here, challenge was that given the NGS dataset of an HCDR3 cluster selection output, identify those sequences within the existing dataset that have the highest affinity antibodies. And so we have 3 clusters here. We provide the affinities of some antibodies, and we ask the participants identify the highest affinity antibody in each cluster. There are 2,500 different sequences in the first cluster, 3.5 in the second, and about 400 in the third. So what we've done here is we're looking at the 3 clusters, 27F, 28F, 47F. The red vertical line for each represents the most abundant sequence as the affinity of the most abundant cluster in each case. We have 2 lines for the 2 clusters on the left. Purple is for all the antibodies, and black is for the developable antibodies. So there's this developability gap where a significant number of clones were lost. This was true for both the 27F cluster as well as the 28F cluster, and in fact much more for the 28F cluster. In the case of 47F, there was no developability gap. And you can see here the best predicted was 9.2 picomole for the 27F, 75 picomolar for the 28F, and 50 picomolar for the 47F. So regarding I've mentioned already about the developability and how the different clusters perform differently in terms of developability. Generally, the cluster predictions were variable in the sense that, for example, 27F and 47F, there were no antibodies predicted by AI which were better than 10-fold the most abundant approach. 28F, on the other hand, there were 7% of the antibodies that were predicted were 10-fold better, at least 10-fold better than the most abundant sequence. Conclusion here is that overall, the predictions were not much better than random picking. Challenge 3, and this is the one that I think most people were most excited by. So over 30,000 sequences were provided from a selection output against the RBD. The affinities of 144 antibodies were also provided, and these were scattered across the different clusters. And the challenge was design a high affinity antibody that was not in the not in the NGS dataset. So this shows here at the top, this shows the experimental SPR distribution. Here we have a ranking of the various organizations and how they performed in terms of developable antibodies, which are open circles, and non-developable in purple. What you can see is that these three organizations were able to produce antibodies that appear to match the best experimental. These were in fact too close to call by SPR. And so we looked at those by Kinexa. And what we found is that the top AI binder had an affinity of 2.9 picomolar, indicated here, which was better than the best experimental, which was 9 picomolar, as measured by Kinexa. The second best had to be disqualified because it was had poor developability, and the final second place had an affinity of 9 picomolar, which was equivalent to the best experimental. So if we look at these out-of-library designs and compare, again, looking at all antibodies by SPR and developable antibodies, you see there is, again, this developability gap. And if we compare the affinity range and developable here for experimental, you see that experimental is providing 24% of the antibodies had an affinity better than 100 picomolar, whereas less than 10%, that was for less than 10%, that was true for AI. And you can see the others range similarly. About 50% of the AI submissions were either non-binders or not developable. So, general conclusions, challenge 1. Up to 40% of the designs were non-binders or had poor developability. The parental did have good developability. A significant number of the models showed a 5 to 10-fold improvement over the parental, with two antibodies, showing affinity improvement over the experimental approach, representing 0.6% of all the submissions. Challenge 2, the number of developable binders depended upon the cluster, with none from 47F with all antibodies from 47F showing good developability, but some of the others as high as 40%. A significant number of models improve in identifying additional picks within a corresponding cluster for a couple of the clusters. However, it's not consistent. And for the novel designs, the greatest number of either non-binders or binders with poor developability were identified in challenge 3, which is the out-of-library design. And only 2.4% of submissions showed improvement over the best experimental, with the winning approach producing a 2.9 picomolar binder, which is really quite remarkable. So to conclude, challenge 1, the best two antibodies derived by AI improved upon the experimental. However, most AI antibodies are worse than most experimental. And using this approach, if consistently effective, would enable a time saving of 2 to 3 weeks over the experimental approach. Challenge 2, prediction does appear to expand hit selection quite well. However, no significant affinity improvement identified. And I would argue that essentially it's no better than just random picking. Challenge 3, de novo antibody designs given over 30,000 sequences and almost 150 affinities. The best AI designed antibody was extremely high affinity, 3 times better than experimental. However, although it passed the developability criteria we had provided, it had low specific activity and cooperative binding profiles. wasn't eluted from the hit column, and so probably was not clinically developable. However, because of the way we framed the competition, we had to give this this was the winning antibody. AI is only better than experimental in a pretty low number of cases. A few companies do show improved distributions, although many have a similar distribution to the experimental. Now, we will be releasing the data set and all measurements and sequences and the source data to the community after the publication to now enable others with similar models which they wish to test to compare their performances to what we've described here. So going back to the objectives, to understand what is really possible, hyperreality, I think it's fair to say that it is reality given good enough data. I should say the detail and volume information provided in these data sets is not usually available until the end of an experimental discovery campaign. And I also believe good results probably depend upon high quality data sets. I don't believe we've hit an AlphaFold moment. There's no definitive winner across all the challenges, although some were quite consistent. Identify specific areas where AI may be of potential value. So I think affinity maturation can add additional leads at time savings of 1 to 3 weeks. Discovery prediction can expand the number of hits within distinct clusters, but again, as I said earlier, not much better than random. And out-of-library design can expand the number of leads, extending the diversity with some providing very high affinity antibodies, but still not as developable as the experimental yet. Regarding the different algorithms, it was quite interesting that the different algorithms performed very differently. We had very distinct winners across all challenges, with one biotech group faring quite well across challenges 2 and 3. And so what's next? Well, we are going to do another challenge. The idea is to really have a bake-off between experimental and AI. We would like the target to be announced by an independent entity such as the Antibody Society, and judge similarly antibodies on affinity and developability, and provide 10 weeks to deliver sequences of 3 top antibodies using any method, in vivo, in vitro, AI, design, test, build, whatever. And then perhaps a repeat of the affinity challenge given an NGS dataset, but without providing any affinities, and also identify antibodies binding different epitopes. Partners involved here, this competition would not have been possible without the really deep collaboration of the partners indicated here. And so with that, I'd like to thank you all and pass over to Dan.
Thank you, Andrew, for that great talk. Today, I'm going to introduce Cartera's HT-SPR technology and go over a few applications. We'll talk briefly about binding kinetics and affinity analysis, describe how we approach epitope binning, and then go into an example where these approaches were used to characterize the humoral or immune response to malaria by 2 publications, one from a group at the NIAID and the other at the University of Oxford. And then we'll talk about polyspecificity characterization and specific activity characterization. The Cartera ecosystem is shown here. We have 3 instruments: the original LSA, the higher sensitivity LSA-XT, and our newest instrument, the Ultra platform, which has even higher sensitivity that allows analysis all the way down to fragment molecules. It's a great platform for biologics as well. Then we have a software environment where we have the Navigator software, which is used to set up experiments and collect data. Then 2 analytical software packages, which are all bundled together. One is called Kinetics and one is Epitope, and it's relatively self-explanatory what those 2 focus on as dedicated software packages. And then we have biosensor chips and consumables that allow you to run all these assays. This high-throughput array-based SPR is really useful in many of the core applications of biologics and therapeutic screening and characterization, those being kinetics and affinity analysis, competition-based epitope binning, peptide and mutant mapping, and quantitation. In addition to having a hardware architecture that makes these assays very high throughput, and not very sample consumptive, we've developed these analytical software packages that make these data processing easy, fast, and also have lots of tools for visualization. The platforms work by having two semi-independent fluidic modules. One side is a 96-channel side that can address the chip surface and flow 96 samples at a time, and then index to two locations on an Ultra for 192 spot array, or 4 locations on an LSA for 384 spots. And then those can move out of the way once the ligands are captured or immobilized to the surface. And the single channel side can dock on and do one injection over the entire surface for what we call one-on-many analysis. So this is where we get the incredible efficiency for things like kinetic screening or epitope binning, where that single injection volume gives you parallel analyzable data across everything that's mobilized onto the array. A common application, one that was used a lot in the AI antibody study Andrew presented, is capture-based kinetics. In this case, we have a capture surface. In this example, it's an anti-human IgG-Fc surface made as a lawn using the single flow cell. And then we capture, using the 96-channel side, the ligands to the array. This can be replicates or titrations of ligands as well to create different surface densities. And then we titrate the analyte over it to get the kinetics. This assay can be scaled very high to 1,152 unique samples in a single unattended run. You could do multiple antigens per antibody in that run if you want. And this is an example of data generated from a run like that. This is a PD-1 binding, 384 interactions, set up in afternoon, run in the evening. We have 8 concentrations of PD-1 being injected, starting at 1 micromolar. And this entire analysis only used 7 micrograms of antigen. See, there's some data flagging from the software. We have gray showing no binding. Kinetic limitations are highlighted in pink. And we have some poor fits being flagged in yellow. The other application I want to briefly introduce here is epitope binning on the LSA platform. This is done by creating an array of antibodies and then using one of 2 assay formats. We typically use what we call classical sandwich binning for monovalent targets, where you immobilize the array of antibodies, then inject the antigen followed by a sandwiching antibody. There's also the premix approach, where you take the same array of antibodies but inject a fixed low concentration of antigen with excess competitor antibody to look for a gain or loss of binding based on how those antibodies compete. Our epitope analysis software has really sophisticated tools for processing these large experiments. Here you can see many different analyte injections occurring, where the antigen injections, then the data is normalized via this bar. There is a report point bar where we are populating the heat map, and then you can set cutoffs to determine things as sandwichers in green or blockers in red. The software will then automatically sort the data based on shared competition profiles to form epitope clusters and dendrograms, and build these network plots where every clone is a node with a connecting line between them, showing competition, and things that are in the same epitope bin or community are colored in these shaded regions. And this is a highly customizable and flexible way of interpreting this data. Okay, with that, we're going to move into an example of how these techniques have been applied. This year, or I guess it was the end of 2024, there was 2 Cell papers that published in the same article on sort of overlapping topics. There's even some authorship sharing between these. One is from the Josh Tan's group at the NIAID, and the other is the University of Oxford, where they're looking at immune response to Rh5 in malaria. Malaria is obviously still a very important disease. There's around 240 million new infections reported every year. So it's a huge disease that doesn't have sufficient treatments out there. And the Rh5 protein is one strong candidate for effective vaccine for the blood stage pathogen. So there's a protein complex that comes off the merozoite pathogen before it infects a red blood cell, and it's only exposed transiently, and it binds to a RBC marker called basogen. This RH5 protein is fairly well conserved, making it a good potential target for a vaccine to end blood stage infection of malaria. So we'll look first at the NIAID paper that was done here. This paper was really interesting in that they looked in detail at the humoral response to malarial infection in a bunch of patients in Mali from a village that is just truly endemic with malaria, very high infection rates. So all of these patients are exposed multiple times a year to the pathogen from the mosquitoes. And they found that the RH5 protein has a sort of rare and transient immune response. So if you look at a newly infected patient, as a couple weeks after infection, they have about 5 RH5 positive B cells per million B cells in their body. And it goes away, drops down to a much lower number quite quickly thereafter. Whereas RH5 vaccinated patients, and these are normal, healthy patients in the UK, have a fairly durable and high frequency immune response to the RH5 vaccine. If you look at the affinity distribution of clones that are taken from either newly infected patients or vaccination, the affinity distribution is not that different, but the frequency of neutralizers is quite low in the identified RH5 natural infection patients, but fairly abundant in the vaccinated cohort. Interestingly, though, 2 of the most potent neutralizing antibodies in the entire study were found in the infected patients. So it is possible to generate RH5 neutralizing antibodies from natural infection, although it may not be the dominant response. And interestingly, those 2 antibodies actually had doppelgangers in the vaccinated set from the UK patients, where there were antibodies using the same V genes and had equivalent CDR links. So there is potential that RH5 vaccination could boost neutralizing antibodies in these patients. The study also looked at epitope recognition of all of these antibodies. They got a very nice description of the competitive epitope profiles, where they have a number of independent epitope regions and some overlapping regions as well. When you look at the growth inhibition relative to the epitope bin, you can see there's sort of 3 main clusters that are primarily neutralizing, and then other epitope clusters that actually do not confer any neutralization. When this is viewed structurally, we have the basogen sort of shadowed. When you look at the top, it's not shown in this view when you're turned away. There's the epitope clusters 1, 2, and 3 that are actually all confer neutralization, and some others that were present that seem to have either very limited neutralization or no neutralization at all. So the idea here is that you could potentially design an RH5 vaccine that is only presenting these neutralizing epitopes for the vaccination. That way, when you boost patients that are being repeatedly infected, you're not sort of driving the immune response to the non-neutralizing epitopes, which seem more abundant in the actual infected population. Okay, so to shift gears to the other paper from Oxford, this is a very similar study where they're looking at just 21 or blood B cells from the blood of 21 patients vaccinated in the UK. They did a B cell identification using flow staining and isolation. They did synthetic sequencing or synthetic DNA production to make expression vectors and get recombinant antibodies produced for 240 antibodies from these 21 patients. they did the epitope profiling as well, and no surprise, it looks very similar to the epitopes that were discovered in the NIAID set. So similar epitope coverage in this set of antibodies. And they looked at bathogen blockade, which again, it's these sort of discrete 2, 1, and 3 bins that had bathogen blockade, and those were the same ones that had growth inhibition. Interestingly, in this analysis, they identified that there were 36 MABs from 4 separate patients that showed very similar Phe gene usage and had really high potency. One in particular set of V genes, the 37136, was the most potent and had multiple instances. If you look at that, it's the one in red here. When they did a germline reversion of the heavy and light chain to those genes, they found that even with both the heavy and light chain substituted back to the germline, they had full maintenance of potency. It's only when they go to the heavy chain knockout CDRs that they lose activity. So this suggests that there's sort of a genome level public clonotype for potent Rh5 neutralization in these, and bodes well for making an effective vaccine. They also use this epitope binning information to analyze possible synergies of antibodies that were non-competitive with each other, pooled together in the neutralization assay. And the dotted lines show what the expected additive potency of these cocktails would be, and the solid lines show the actual observed behavior. And in some cases, they saw up to a 45-fold increase in potency by mixing these different clones together. And some of these actually included clones from bins that were non-bathogen blocking and not neutralizing on their own. So this sort of reinforces the idea we see repeatedly throughout the literature that binding multiple antibodies to these pathogens in different epitopes is a fairly effective strategy for neutralization. And there's good opportunity for cocktails or consideration of these various epitopes in vaccine development. Okay, now we can move on to 2 other applications. First, we'll talk about specificity and polyreactivity. Obviously, the specificity of a drug is critical for its success, and sometimes polyspecificity, meaning binding to different targets, can be desirable. You may want to neutralize different species or isoforms or family members of protein, but unintended polyspecificity can also decrease efficacy or increase toxicity. So it's important to have assays to assess this and to understand that. Polyreactivity, on the other hand, is typically referred to as nonspecific binding, is the tendency for binding to common chemical features like charged domains or hydrophobic pockets. And candidates that are polyreactive often demonstrate poor PK, higher levels of toxicity, and often result in clinical failure. So high-throughput SPR is a great way to characterize both specificity and polyreactivity in large automated assays. We have 2 sort of published examples of this. There's a Cartera app note using the capture of histag proteins for polyreactivity profiling. It's described here. And then there's also a peer-reviewed publication that we did with a group at Specifica, looking at the mobilization of biotinylated proteins to the array and the injection of antibodies over those to profile their polyspecificity. I'll go quickly through that example here. We used a 96 well plate set up with duplicate titrations of 7 polyspecificity reagents. So we cut a broad range of surface densities. We had DNA monomer, polyglutamic acid, ganglioside, HSA, BSA, and ovalbumin, and biotinylated fibrogen. So these were immobilized to the array on a streptavidin chip, and then, or a neutravidin chip actually. And then we injected the antibodies at 250 and nanomolar and 2.5 micromolar to look for the polyspecificity. This shows an overlay of sensorgrams from the assay. You can see we have two antibodies that had high levels of polyreactivity. Shown in blue is the lenzilumab, and green is gantenerumab. So you can see we get lenzi binding to BSA, monomer, and polyglutamic acid, whereas gantenerumab bound to fibrinogen and ovalbumin. If we look at the analyzed data for this, we see that we get a profile that shows clearly sort of what the background is of these various antibodies, as well as the ability to see the increased polyreactivity. You can use this to make cutoffs and determine what you would think would be a problematic level. If we zoom in on one of these, like fibrinogen, that had a really high response for one, it makes it a little hard to see what the sensitivity is to the others, but we can zoom in and omit the highest one and look and see that there actually is a very low background generally, and that a number of other clones also have above background levels of polyspecific binding. And so at 2.5 micromolar, this assay seems to be quite sensitive, similar to what you see in ELISA, except these assays can be run in an automated fashion at a large scale. Okay. And the last example I will go over is specific activity analysis. So biologic therapies are complex molecules, and there's a lot of possibilities for different levels of activity of them. You can have differential glycosylation, aggregation, things like deamidation, oxidation, folding issues. With antibodies, you could have light chain pairing issues, all which impact the potency or the activity levels of these preps. Specific activity assessment can be seamlessly integrated into kinetic screening, as was done with the AI antibody study. And it compares the mAb capture level, so how much antibody you're capturing to the array surface, to the Rmax in the kinetic fits, the maximum binding level observed. So there's a feature in the kinetic software that makes this really easy. We look at a report point of how much mAb we captured, and then when we fit the data, we end up with an Rmax term, and we can compare that to a theoretical Rmax. So the theoretical Rmax is just the molecular weight of the analyte times the ligand valency divided by the molecular weight of the ligand times the immobilized ligand level. So it's just a stoichiometric expectation of the signal. And then the percent activity is just the experimental Rmax divided by the theoretical Rmax. So this can be a useful tool to compare the relative activity of different clones, and if you're screening diversity, it can also be used to assess things like new cell line development or purification strategies. If you have batches of things that should be very similar, you can capture them with fairly high N into the array and do this analysis and see whether there are any differences in activity. To actually complete this, you can input the molecular weight and the valency in the ligand table in the analysis software. So you can have different sets of antibodies with different molecular weights and valencies. We input the molecular weight of the analyte. We do a report point analysis and set it as the capture level to say how much antibody is on each spot in the array. And then when you fit the kinetics, you check the show specific activity box, and it will make a column where it's comparing the Rmax from the kinetic fit, that maximum binding level, to the theoretical Rmax based on that capture level and the stoichiometry and molecular weights of the compounds. If we look at how this was applied in the AI antibody challenge, this is the specific activity of all 540 clones. Most of them had relatively good specific activity with greater than 75%. But there was also a number that had very poor specific activity with less than 25%. These were actually excluded from the running in terms, because these are not properly behaved antibodies. And if we look at the comparison of the capture level to the specific activity over this entire data set, we see that there is a slight trend towards at higher and higher capture levels, you get a bit lower specific activity. This is typically expected because you can have some crowding effects at very high density. But in general, this is a clone and sample specific phenomenon. So each clone is actually present 3 times in this at different densities. So that's kind of why we have these clusters. So it is an innate property of the ligand you're capturing, how much activity is there. And so this is no extra steps to do this analysis. If you're running capture kinetics, you already have all of the data in the analysis software. You need to do this. It's just a matter of checking the boxes and filling in the fields and evaluating that. Okay, so in conclusion, high throughput SPR on Cartera platforms enables a broad understanding of the nature and specificity of these humoral immune responses with both affinity and kinetics analysis, as well as high throughput epitope binning and comparisons. You can compare polyreactivity of many clones to many targets easily in large automated assays. And you can assess the sample specific activity levels as part of a normal kinetic screening and characterization workflow. And with that, I'd like to thank all of the excellent scientists who worked on all of this data that I presented here today. Thank you. And we can take some questions now.
Thank you everyone for your attention, and thank you Andrew and Daniel for your incredible presentations. We'll now move on to the Q&A session. Feel free to submit your questions using the box on the right-hand side of your screen. Just type the questions in and click send. We'll do our best to address as many questions as we can. We've got just over 5 minutes left, so we should be able to get through a few questions. But don't worry, any questions we don't get to will be answered offline as soon as possible, and you can keep asking questions even if you're watching this webinar on demand. So we've already had a few questions, as I've said, so let's just dive straight in. I think, Dan, why don't we start with you? This one seems tailored to you, which you kind of touched on at the end there. Is it possible to integrate polyreactivity testing into typical kinetic characterization assay setups? If so, how would you go about doing that?
Sure, yeah. Typically in a capture kinetics format, which is the most common way these things are applied at scale, you can either include a separate capture of the antibodies and then injection of these reagents that we showed immobilized in that example, which was sort of a dedicated polyreactivity assay to see how much binding to the captured antibodies you get. You can also do that just, sort of at the end of a titration experiment, do several injections of different reagents as well. So there's ways you can pretty seamlessly integrate that into normal screening workflows without much extra sample prep or time. Thanks.
Fantastic. And then another one I think here for you, Dan. And how does having low specific activity impact the interpretation of the kinetics of the ligand?
So they're a bit independent parameters. Sometimes if a antibody or a protein has, some aspect to it to decrease its activity, it can have heterogeneous kinetics where you won't get a one-to-one profile because there's different affinities being modified, whether it's a deamidation site or something. Many other times you will get just a smaller version of the sensogram. There is a certain component that is totally inactive in the sample, and then so you just manifest as less binding for a given amount with what appear to be one-to-one kinetics. And that's probably more common, or maybe they're equally common to have both those situations.
Fantastic, thanks for that. Again, we've got a few minutes left. So Andrew, I think these ones are more tailored to you. A little bit bigger picture. Do you think AI will ever replace experimental methods to generate antibodies?
So that's a really good question. I'm glad at this end of my career, as opposed to the starting end of my career, I think that experimentalists will always be required to confirm the AI prediction of antibodies. I think it may happen. There are certainly publications at the moment that claim to make it happen, but I just don't know. This is one of the reasons why I started the challenge. There was so much out there about how AI was going to take over antibody discovery, as is claimed for the rest of the world as well. I just wanted to understand what was real. The next challenge that I mentioned in my talk is going to be a real bake-off between AI in vivo, so immunization of mice, and in vitro, and we'll see how the AI algorithms fare. I think the only way you can really assess whether AI is going to work is by doing benchmarks of this kind. I don't think you can rely on statements coming from self-interested parties that have a lot of money at stake.
That's fantastic. Again, I think that's very accurate, an honest assessment. I like it. So then the next one we've got, again, another big picture one, Andrew. Seems like a good one to wrap up on. How do you envisage the next AI antibody competition? Who will decide the target and how will it be judged?
Right. So we at Specifica would like to participate in the next one. We didn't participate in this one because we felt that there might be accusations of sort of insider trading, as it were. And so what we're talking with the Antibody Society, which appears to be keen to take on parts of the challenge, and in particular identifying the target. They will also be involved in judging and helping analyze the results as well.
Fantastic. Hopefully that interested a lot of members of the audience. Hopefully they're fully aware of, and we'll be able to keep an eye on that in the future. Again, unfortunately, that is all the time we have today. Please just remember that any questions we didn't get to will be answered offline as soon as possible. And you can continue to ask questions even if you're watching this webinar on demand. By attending today's webinar, you'll automatically receive a certificate of attendance that will be emailed to you using the email that you registered with. No need to request it, it will arrive shortly. Once again, I'd like to give a big thank you to everyone for paying attention, and an incredible thank you to Andrew and Dan for taking the time out today to talk to us. Thank you very much. It's been a pleasure.
Thanks very much.
Thank you.
Bye.